initially to be supportive cells. They contribute a lot to the structure of the brain. They participate in the blood-brain barrier. They can regulate the extracellular environment of the neurons. But in addition, they also can regulate a lot of neuronal function. They can regulate synapses. They can respond to various peptides and hormones. And they can also secrete steroidal hormones, which I'll show you. So they isolated cultures of neurons, oligodendrocytes, and astrocytes. Then they exposed them to pregnenolone, which is the immediate precursor to progesterone. And then they measured the progesterone production. And we see that the astrocytes are the most productive in terms of progesterone. And I didn't show this, but they're actually the most productive in terms of steroid production in the brain. So therefore, our studies focused on astrocytes, specifically hypothalamic astrocytes, in order to produce the progesterone needed for the LH surge and positive estrogen feedback. So now that we know we need estrogen and progesterone in order to have estrogen positive feedback, but we need to know where is this estrogen acting? Are there estrogen receptors and astrocytes to actually carry out the signaling for estrogen? A study in 2004 by Victor Chavan basically took hypothalamic astrocytes, then lysed them, and then ran them on a western. So in B, you have the cytoplasmic and nuclear fractions. And on the western blot, in lanes 2, 3, and 4, lane 1 is actually a control. So we see estrogen receptor alpha exists in lanes 2, 3, and 4, are samples. And then estrogen receptor beta also exists in lane 2, 3, and 4, which shows that there's estrogen receptor alpha and beta in the nuclear and cytoplasmic fractions. Then they isolated just the plasma membrane fraction. And then again, ran a western and probed it with estrogen receptor alpha, which we see here, and estrogen receptor beta. So this shows that there is actually estrogen receptor alpha and beta, not only in the nucleus, but also in the plasma membrane. So this is somewhere where the estrogen can act. So there's evidence goes back, where suspicions for membrane estrogen receptors go back to the 1960s with electrophysiology studies. The main reason is that in those studies with estrogen, they give a large electrical response. In astrocytes, the main axis of response is an increase in free cytoplasmic calcium. When these cells are exposed to estradiol, they respond within an order of seconds with a large release of intracellular calcium. So calcium is sequestered in the smooth endoplasmic reticulum. And then so when estradiol signals, it actually releases the calcium from the endoplasmic reticulum and causes a free spike in calcium.
John, estradiol can dissociate very easily from albumin. We and others have shown that many, many years ago. And I'm wondering, when you add that, how much do you think, have you done some experiments where you've just added the albumin-bound estradiol and then seen with assay the free form and the bound form? Because that would be critical to say the action is purely in the membrane, or someone has gone. Has anyone done it? You may not have, but
so this study, this study I did not work on.
No, but there was a paper. Information.
Right. No, there was a paper in 1999 by Stevis, who basically, I think, took E6-BSA and E17-BSA, and it showed that there was, when you first dissolve it, there's actually free form of estradiol, and then they used a micron cartridge and they used a filter, and the filtrate also had free estradiol. But then if they took the retentate, and then they remade the E6-BSA, that there was actually no free estradiol in that. And then, so I believe on these experiments, they actually did do that. They actually filtered it and then used the retentate. And I think they also used the filtrate, if I'm not mistaken, and there was no response on that filtrate. But there's also other evidence for membrane estrogen receptors that I'm going to show you. So this is just the beginning. I'm giving you some background before I so estradiol has a rapid free calcium response in the hypothalamic astrocytes. And if we use E6-BSA, which is basically estradiol that's conjugated to bovine serum albumin, which is a large molecule, it's membrane impermeable. So therefore, if you have a response, that usually means that it's reacting to something on the plasma membrane. And with E6-BSA, we actually also get a spike in calcium that is very similar in magnitude. So this suggests that the estradiol may act on a plasma membrane estrogen-associated receptor. In addition, if you take hypothalamic cultures and you incubate them with estradiol, you actually get synthesis of progesterone. Again, evidence goes along with that the astrocytes can produce progesterone. Now, if you give the same cells thapsigargan, and thapsigargan is a calcium ATPase inhibitor, so normally calcium is sequestered into smooth endoplasmic reticulum. But if you give thapsigargan, it actually allows the release of this calcium, and the calcium spike is very similar to the estradiol. And then in addition, after thapsigargan, if you stimulate with estradiol, you actually don't get any more response. So it tells us that the estradiol is acting on the smooth endoplasmic reticulum to release the calcium. And then thapsigargan alone, without estrogen, can actually increase the progesterone synthesis. So this kind of tells us that if you have a large release of calcium alone in these cells, this is enough. It's not specific to estradiol. So this brings us to the working model of estradiol regulation of the LH surge and estrogen positive feedback. So in this model, estrogen that increases with the maturing oocytes will act on an interneuron. This is, we believe, a kisspeptin neuron. And the estradiol will result in transcription and translation of progesterone receptors. We know this genomic response takes some time, so the estrogen acts on this interneuron to produce the progesterone receptors. And then the estrogen also acts on hypothalamic astrocytes. And we believe it acts through a membrane estrogen receptor, which I'm going to go over in this talk. So we're going to try to clarify which receptor this is. In addition, there is some signaling that will signal through phospholipase C, produce IP3, which then has IP3 receptors on a smooth endoplasmic reticulum, and that releases the calcium. And then the calcium that's released will activate various kinases, such as protein kinase C and protein kinase A, which results in phosphorylation of enzymes. And then the enzyme activity, we believe, increases, such as P450 SEC, STAR, and possibly pregnenolone. 3-beta-HSD, which will increase pregnenolone and progesterone synthesis. And then at the high levels of estrogen and progesterone, it'll stimulate the interneuron together to release and stimulate GnRH neurons, and that releases a surge of GnRH to stimulate gonadotropes in the pituitary to release a surge of LH to trigger ovulation. Now, from hippocampal and hypothalamic neurons, they've demonstrated that the estrogen receptor can actually signal to a glutamate receptor. So therefore, in my studies, I wanted to see if this is also true in hypothalamic astrocytes. So from here on out is all my data. So just wanted to let you know. And so in my system, we have a gravity perfusion system where we perfuse astrocyte cultures in media and then give them boluses of estrogen or other drugs. And when you give them estrogen within 10 seconds, you'll see an increase in free cytoplasmic calcium. So the first thing we did is do a dose response curve for calcium increase. So we see that at 0.1 nanomolars of estradiol, we actually get a significant increase in calcium. And then by 1 nanomolar, it's actually a maximal increase because at 10 and 100 nanomolars, the response doesn't increase any further. And so this is actually physiologically relevant. The peak levels of estradiol in the rodent prior to the LH surge and estrogen positive feedback is in the hundreds of picomolars, so it's somewhere in between these two. So these are relevant ranges where the calcium is going to go up.
When you present data like this and you have your sample size, is this, are you imaging a single astrocyte and, or is it, so is that?
These are, so 25 individual astrocytes.
And how many cultures were created to produce that 25?
It typically takes about, I mean, it ranges, but at best it takes about 2 or 3 runs, so 2 or 3 separate cultures. But sometimes if they don't respond well,